So I'm going to give a relatively brief discussion about methods for trials when one is trying to develop treatments for disorders from the point of view of the challenges that one faces specifically around subject recruitment and number of subjects that are available. So first, here are my disclosures. And, you know, the challenge is that the orphan disorders are by definition rare, and their being rare means it's going to be hard to conduct studies. It will present some specific challenges to conducting studies evaluating treatments that you want to be able to develop for these conditions. So I'm going to talk on strategies for minimizing the impact of the size of these populations on a capacity to successfully carry out treatment studies. The two things I'm going to focus on are challenges in recruitment and challenges in signal detection, mainly due to limitations in how big study populations are. So that means how big a trial can be at reasonable cost. Really going to focus on the second point because just have more to say on that topic. So just in reflecting on this, it became very clear to me that when I work with trials of disorders where the population is very small, there's really only one effective way to recruit people, and that is to go to the clinics where these people are treated. Usually, there are specialized clinics in university settings where, and sometimes in private practice, where people with specific conditions are managed. And this, the ability to do this depends on whether the condition is episodic or whether it is a lifelong condition. So this really works best for conditions like narcolepsy, where I had a cohort of patients with narcolepsy over the years that I've put into trials, and I've kept a waiting list of these people, and so that I could contact them every time a trial was needed. If you didn't have that, it would be very difficult to find people due to the rarity of this problem in the population if you're just going to advertise. And with narcolepsy, people, once they get diagnosed, they have the problem for the rest of their lives. So they're generally connected to a practitioner who manages them. And fortunately, well, unfortunately for the people with narcolepsy, but fortunately for the ability to do trials, a lot of people who have the condition are not happy with their ongoing treatment for one reason or another, and are often willing to and interested in participating in a trial of a new intervention. So, this strategy means that you have a set of sites around the country that you identify as the experts in an area, and they have a database of people that they contact, and it's really the only way you can get studies of reasonable size done in reasonable time at reasonable cost. I think another thing that I've used that has been maybe the most useful is that it's important that when you design a study that you do so in a way that people actually receive clinical therapy. And by that I mean not their clinical therapy in the usual sense, but I mean treatment. From if we do a straight up double-blind randomized placebo-controlled trial, patients who have conditions that they're suffering from should and often do ask the question, why should I participate in this study? I have a 50-50 chance of getting placebo, and I'm not going to be able to get access to this treatment afterwards. There's no incentive for me to do it. And so what I tend to do is put in a phase after the double-blind placebo-controlled trial phase, which is a long-term open label phase or something like that. And that way, every single person in the study gets study drug. And particularly if, depending on the nature of the condition, you would hope to make it as available to them for as long as possible, if they choose to. Of course, if they, don't want to participate in that, that's fine. But that way, I found that I can, in good conscience, say to a person, if you participate in this, you will get treated with this, and it is an option to get a kind of intervention for a period of time that you've never had availability to before. And found that that really matters. And then another point that is very effective or important is how much you pay people. When I started out doing clinical trials in medical conditions in my career, we never paid anybody, and we uh, and and I remember and we people were in these studies because to do it to help society, to help better understand and treat the condition maybe that they had. But there was not like, the fact that they, half of them were going to get placebo meant there was unlikely to be direct payoff for them, and might be, might not be. And also the treatments are usually not available for people, as I was just reviewing, after after they're in the trial. So it's not, it's, it has to be an altruistic motive. And that actually was not a problem. I was able to recruit people for studies reasonably well. And then at some point, the culture changed, and people would, like, when you say we're doing a trial, and they would contact to participate, the first thing they would say is, "How much are you paying?" And it's become very clear that that has now a substantial impact on our ability to recruit people and their willingness to be in studies. And it is important that you pay market rates for study participation to people. And the more you pay people, the more success you have in recruiting. And it's sad, perhaps, but it is a reality. And so if you have an orphan condition and the population base is small, and you need to do a study of decent size, then this is a critical ingredient to be able to be successful. Now, I want to move on now to a, a sort of the next phase of things, and that's to talk about the detection and signal challenges. Signal, just the challenge in signal detection. And what I mean by that is that if you assume that there is an effect of a treatment versus placebo, will your study be able to detect it? Hopefully, it's evident to everybody listening that in order for a study to be able to detect a signal, the population of the study has to be large enough for there to be adequate power to show a statistically significant effect. And um, and it's it's expensive to do. The larger the study, the more expensive it is. And in the orphan conditions, it may not be even possible to do studies larger than a certain size. And the effect size you may want to detect may make it impossible to be able to have sufficient power to do a study. So it's cost and feasibility, and that drives considerations of how do you do a study that allows you to effectively detect the signal if it's present with a smaller population. And there's a number of strategies that are out there. I'm going to start with one that I've used that is probably not one that is in the textbooks or commonly talked about, but it's the use of biomarkers. Now, uh, many of us are interested in using biomarkers in studies for a variety of reasons. And but I'm going to focus on this particular one about the potential benefit for signal detection. So what we usually do in many conditions is use a clinical endpoint, um, like, let's say if it's depression, a Hamilton Depression Rating Scale, or if it's narcolepsy, let's say a Epworth Sleepiness Scale or something like that. And a challenge with those is that there's a lot of variability built into them because they are judgments of people, either a clinician or a patient, that are very complex. And we've tried to make them as standardized as possible. But by the nature of a rating of this sort is just very, um, you know, very, very hard to standardize and to make uh, a very sensitive indicator of conditions. And because of that noise, the studies with clinical endpoints are thought to have vulnerability to false positives that are occurred due to nonspecific effects or bias. An example would be that you accidentally you have a statistically significant effect, but the reason you had it is because you accidentally happened to randomize people to one group versus another group that that had a specific condition. They had more severe illness, or they were had a certain trait that led them to rate their condition as more improved, or there's a variety of factors. It could be investigator bias. There's often incentives for um, for showing effects, and if you have uh, you have a couple of sites and they have a lot more people on drug than placebo, uh, that bias can change. And you have one investigator, let's say, who has a huge bias, and they have that can lead to a false positive. There's so many reasons why a study could have a p less than 0.05 difference between a drug and a placebo, and it's not because the drug has greater therapeutic effects than placebo, actually, because of all these other things. And so, and the clinical endpoints, just because of their variability and and sort of the I don't know if the inaccuracy isn't the right word, but the fact that a human judgment is involved makes this, is some believe, make them more vulnerable. So biomarkers um, have the potential benefit, if chosen properly, that they could be way closer to the biology of what a drug does than a rating of that sort. So imagine you're looking at a condition that, let's say, or treatment, excuse me, that binds to the serotonin 2A receptor. Um, it's a long distance from binding to the serotonin 2A receptor to somebody reporting how sleepy they are. That's a long biological distance, if you will. However, if you looked at the parts of the brain where there are serotonin 2A receptors, and you asked, is there a change in blood flow in that area of the brain? And your hypothesis is that the serotonin 2A receptor binding that the drug does changes blood flow, um, because it alters activity in that area, and that altered activity mediates the clinical effect. Then what you're measuring could be pretty close biologically to effect the drug is having. And so some have hypothesized, there's a number of publications that have argued this, that you could potentially do studies with smaller populations, particularly in earlier phases, when you are trying to determine, is there something here of value, that you could do that using a biomarker that's part of your hypothesis about how a drug works, that's relatively close to the clinical, or excuse me, the biological effect of the drug, and 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 cut down the sample size and be more efficient. Now we have a big challenge. When you work with DSM disorders, we have limited capacity to do biomarker development. So, for example, if I said, um, I'm going to work with disorder, uh, schizophrenia, well, there are all kinds of hypotheses about where in the brain, an antipsychotic treatment might work, or some kind of treatment might work to mediate schizophrenia. But our weak understanding of the neurobiology of schizophrenia itself makes it very difficult to have a convincing hypothesis about what happens between the receptor effects and the improvement in schizophrenia you're hoping to see, uh, because of our limited understanding. RDoC, this is the Research Domain Criteria project that was started by Tom Insel at NIH and Bruce Cuthbert at NIMH, is dimensional by nature and linked to biology by nature. And therefore, if you look at this RDoC matrix, which is essentially a competing framework for diagnosis and outcome assessment, that there are entities that are, many of them have biomarkers associated with them that could be used to measure them, and of varying degrees of validity um, and and uh, and ex use experience with use in clinical trials. Some are more there's more evidence showing they can detect signals in trials, and more showing that they're valid. But others, where that's lacking, just like with the DSM, it's quite challenging. But there are some that exist, and working in this framework, or at some framework that allows more biological biomarker potential, can then make a big difference in being able to improve efficiency of study design. And just a little bit further thought about this concept. Um, the biomarkers, uh, if you to use them effectively, they really should best be employed in a proof of mechanism kind of framework, a structure that that I'll describe in a second. Um, but but uh, what but this is important because you can't just take any biomarker and stick it in any study and just and then have a robust capacity to lower sample size. Really, what you can have with a biomarker is both so smaller sample size and at the same time some safeguard against carrying out the super expensive studies, that when adequately controlled with clinical endpoints that needed to show effects, uh, and also to address to some degree the likelihood that you have a trial that's positive, but it's due to nonspecific effects and bias. I realize I've said a bunch of stuff without explaining it. I'm going to try to explain it now. So um, there's been articulated some methodology about how to do rigorous proof of mechanism studies, and um, and that is employing a biomarker as primary outcome. So before you can do the study, it's important to have some way to measure what the drug is doing at its neurobiological target. An example would be being able to do PET scanning to assess the degree of receptor occupancy is the first step in the process, because that will tell you need to be able to choose a dose of drug that robustly engages the target. And so in this case, let's say it's serotonin type 2A receptor antagonism, you need to make sure you choose a drug that robustly blocks the serotonin receptors. Once you have that, then you do a proof of mechanism study with the biomarker, testing the hypothesis that engaging that target, let's say blocking the serotonin type 2 receptor, has a specific effect on the brain that is thought to mediate the anticipated effect. Let's say increasing activity in the neurons in the dorsolateral prefrontal cortex. And that's a specific hypothesis, and presumably there are 5-HT2A receptors that affect that are thought to affect that function in that region. And so you're basically walking back from the clinical to a closer to the biology uh, place that's sort of intermediary between the biological effect of blocking the receptor and the clinical effect. And the idea here is if you have a model about how the drug is supposed to work in the brain, and you can't show that model is correct, you shouldn't go on to a study with a clinical endpoint where if it's positive for any reason, under the sun. But if it's not positive because it does what you think it does in the brain, your model is wrong and you need to go back to the drawing board. So this structure is meant to sort of provide a way to have an extra step in the process in a way that safeguards against doing a study that's riskier, and which is critical for working with a population where it's extremely expensive to get an adequate sample size. You don't want to go to that next study. But otherwise, what we normally do is a phase 2A or an early phase study with a clinical endpoint that's horribly underpowered, that is extremely risky. That has been the way we have proceeded for years and years. And so this strategy provides an opportunity to safeguard a bit. So the idea literally is, if you establish proof of mechanism, so let's say you give a person a 5-HT2A antagonist and it changes dorsolateral prefrontal cortex neuronal function as measured by EEG or by an fMRI or an MRI or something like that. Then you you said, yes, it looks like what I thought was going to happen and should happen, that I think mediates a clinical effect, did happen. Then that decreases the likelihood that if you show a positive effect in the clinical and trial as adequately powered, that it's due to those some non-specific effect. It's much more likely mediated because you have achieved the change in the brain that you hypothesized would be there. The other part of it is that our field suffers from the problem that we get negative findings in our phase 3 big trials, and and we don't know why. So the idea is you get a negative study in a phase 3 trial, and then you say, I think the dose was too low, or I think, whatever. You have some issues like that. That negative findings mean, that your model was in fact wrong. Engaging that area of the brain does not in fact have the clinical effect that you thought it was, and you have to go back to the drawing board. But it's better, that you learn that and you establish that than try to continue on with a program where you think, oh, well, I think my model's correct. I think the dose was too low or something like that. So, or I should have gotten a more severe population, or I should have gotten a whatever. So I'm just now going to give an example of a study that I did that is in this framework and just illustrate sort of the steps involved, and hopefully this will be of help to people. So I did a study, a proof of mechanism study, to assess whether kappa opioid antagonist drugs. There is a kappa type opioid receptor. The mu one is what everybody knows about, but the kappa receptors are just another type, and antagonists of it have a very large basic science literature supporting that they have potential as treatments for anhedonia. Basically, in animals, in a variety of contexts, that blocking the kappa, that first, dynorphin is released when there's stress, and it binds uh, to receptors called kappa opiate receptors, and they diminish the release of dopamine um, into the ventral striatum, both at the level of the ventral tegmental area and at the level of the ventral striatum. Nucleus accumbens, I'll use that word just to not confuse people. Nucleus accumbens is part of the ventral striatum. And we all know, hopefully, that the nucleus accumbens is like the central area we believe mediates reward. And so the animals show clinical signs of anhedonia in terms of not pursuing treats, not, lots of activities that have been classically identified as marvels of this. And so what we did is the first study in humans looking at whether there was a possibility that this had potential. And to do it, we tried to think, okay, if the drug blocks kappa opioid receptors, what in the brain do we think mediates the effect, the clinical effect? And it is increasing activity in the nucleus accumbens, increasing through dopamine release. We didn't test the dopamine part of it, but we tested specifically the activity in the nucleus accumbens. And you can see here in the figure that you have the dynorphin blocking it. When it binds to this kappa opioid receptors, it hits the dopamine neuron both at the level of the ventral tegmental area and the nucleus accumbens, and inhibits its release of dopamine. So we did an 8-week double-blind parallel group placebo-controlled study with a specific kappa opioid receptor antagonist, J&J 67953964. I will never forget that number, even though it's now irrelevant, because this drug now has a name. It's called aticaprant. And we looked at 10 milligrams versus placebo. This was a drug that was owned. Has a J&J number for Janssen, or Johnson & Johnson, excuse me. And but it was owned by Lilly at the time, and Lilly had shelved it. And they said, we're not interested in this drug because we tested it for something like some alcohol use disorder or something, and it failed the trials. And at that point, people were very nervous about doing repurposing drugs because so many trials were failing, and they just said, we're done with this. It's just sitting there. So I had a grant from NIH to do a study related to this. It's complicated. I won't go into the details of it. But anyway, I approached them and they said, sure, if you want, you can use this drug. And it had already the critical ingredients in place. First of all, we knew toxicology had been done. Its single ascending and multiple ascending dosing had been carried out. And it had a great adverse effect profile, but it was a specific drug for kappa opioid receptors, very potent and specific. And there was a PET ligand that could use to measure receptor occupancy. And it turns out that the 10 milligram dose had already been established to have a robust binding at the kappa opioid receptor. At trough, it was 90% occupying the kappa opioid receptor. That's what you want. That means this dose engages the receptor. We know it will. There'll be no doubt about it. And then we do a proof of mechanism study that tests when you engage the receptor, does it do what you expected it to do in the brain? If we didn't have that PET information, we would have had to have done a PET study to do that, because otherwise you can't be confident that your proof of mechanism study will actually address the question. So we had a clinical measure in the study, the Snaith Hamilton Pleasure Scale, but it was not the primary outcome. It was secondary. And we did fMRI, functional magnetic resonance imaging, during a task called the monetary incentive delay task, which has been used by lots of people, where people are given a cue that they're going to have the opportunity to win money or lose money, and we actually pay them based on their performance in a simple reaction time task. And you average the blood flow, the bold signal, as an indirect measure of neural activity in this area, the nucleus accumbens, during the period where they're told that they have an opportunity to win money and before they know whether they will get winning money or not. And we targeted an effect size of 0.5 with an N of 90, which is a reasonably sized phase 2 study. But in this case, we thought it should be adequately powered if the effect size of this is 0.5. So I'm going to just jump to the chase and show that we had a very significant effect on the ventral striatal activation during that, in anticipation of reward. And the effect size was 0.57. We also had a significant effect on the clinical endpoint. Now, that was not critical for our study, but what was critical is we wanted to measure how big is the effect size on the clinical outcome. And it was actually bigger than I expected it would be, but still smaller than the biomarker. It had an effect size of 0.44, somewhat reinforcing the idea that the biomarker being closer to the direct effects of the drug give us a bigger, likely to give us a bigger effect. This is just shows the clinical effects, and that the drug robustly led to this improvement over 8 weeks. And we saw the other. I'm just going to point out here that every measure of the nucleus accumbens activity was also referred here as ventral striatum. I apologize for going back and forth between these. Was significant, and almost none of the clinical measures that were in the study were significant. This was all across the board, again reinforcing this idea. And some of the effect sizes for the imaging measures were actually over 1. So this we didn't look at anything except the nucleus accumbens. This was very focused. It wasn't like we did hundreds of analyses and one turned out to be positive. So I want to stop there, just showing an example of a proof of mechanism study with a biomarker that has the potential to lower the number needed to figure out if there is something promising going on. And it also is a way to safeguard against sort of the bias effect of doing that may creep in with clinical endpoints, especially if you're limited sample size. And the idea here is, having done this, we have some assurance that if there is a positive effect in a clinical trial done for phase 3, that it would be less likely to be due to nonspecific effects. Just to tie the a bow on this story. This drug, it was with Lilly when I started the study. Janssen had it at the end, Johnson Johnson, uh, and they are now developing it, and it's in phase 3. Based on our study, they went ahead and did did the clinical trials. They had positive clinical trial with clinical endpoint in depression, the anhedonia, and um, they are now doing their phase 3 effort with the drug called aticaprant, and hopefully we'll be hearing more about it. I want to now move to another way to increase efficiency, and that's to use adaptive design. There's a number of designs that are out there that lots of people write about and talk about, but we don't use that often as a way to make trials more efficient, and they can be smaller. And there's a whole family of these, and I'm just going to talk about them in a very superficial way. But uh, the one of the most common of these, um, well, basically what they can allow you to do is decrease the number of subjects you need, and therefore allow you to have better signal detection versus cost ratio. So one of the most common is a two-phase adaptive design. And what that means is you have a first phase where you like do some comparison of drug and placebo, and maybe several doses of drug, maybe one dose. And then you have a stop the trial, and you have an interim assessment, and you do something like you look at whether one of them is looking very promising, and then you drop the other ones and proceed with the trial with the rest of them. And I mean, you drop the other ones and you proceed with the best one. And you may say, well, that sounds like cheating, isn't it? It is. But the way this is addressed, and this is rigorously worked out and accepted by FDA, is that you pay a penalty in your power in your statistical analysis for doing an interim analysis. There's a fixed set way to do this, which is very interesting. I don't have time to go into it. But there's many ways this is done. I'm just going to talk about to add the dose or play the winner. So the add the dose one is you do one dose versus placebo, and then you stop and you do an interim analysis after collecting a fair, decent number of people that you believe is going to be representative, and that's a challenge in itself. And if that dose is not doing as well as you would hope or hypothesize, you add other doses. That prevents you from doing the whole study with say 3 doses. Um, and and also you have some idea about how to power that last half, which you can also we'll come back to this. You can also decide to stop at that point. The more common design is um, the play the winner. You have 3 doses against placebo. You stop after some point, and then you go forward with the best one, or and if none are good enough, then you stop. And which is the futility analysis idea, and which allows you to stop. And the advantages of it is if you're working with an orphan disorder and the studies are extremely expensive because it's so hard to recruit, and then it's a bit of a risk uh, protector, because you save the money of by stopping when you have futility. Now, futility isn't perfect; it's an estimate. It involves calculating the probability of rejecting the null hypothesis at the end of the study based on the data you've collected so far. It's conditional, so it's Bayesian statistics based generally. And if the probability is so low that you think this is not worth pursuing, then you stop it. I've seen a number of trials done like this. I was part of two studies done with deep brain stimulation for depression that were stopped for futility. And that was put in for ethical reasons, and also cost, but mainly ethical reasons. And what does that mean, that like the probability of rejecting the null hypothesis at the end of the study is so low that you wouldn't proceed? Well, what you may sometimes find is that your placebo is doing better than your drug, and it's doing a lot better. If it's doing far enough better, you can be pretty confident the odds of it showing the reverse effect with adding the additional subjects is negligible, or let's say less than 5% or whatever, and you set some limit like that. You can also add some things if you're worried about things like missing data, or that there might be an event rate that's so low that 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 you might not be able to support conclusions. You can do an interim assessment of that. What I mean by that is statistical missing data is obvious, but event rates being too low. For example, let's suppose you're doing a study of suicide, depression, and you're looking at suicide, and no one in the study committed suicide at the interim. That makes it really hard to justify continuing. Um, and let's see. Another issue that's very important for thinking about its design efficiency is trying to control the nature of the placebo effect. In some areas like mood disorders, this has been a hugely impactful issue, and people have struggled with it greatly. You have some studies where the placebo response rate is 50%. How are you going to beat that? And that has happened literally in clinical trials. I think there's some that are over 50%. That's just devastating and expensive. And so people have tried to take some steps. I'm just going to mention one. And this is the sequential parallel control design. And boy, does it look crazy. But the simple thing of it is that you have a first stage where you give people a drug and versus placebo. In this case, it's buprenorphine, samidorphan, a combination which was being used for depression. They looked at 2 dosages in placebo. The key here is that you take the stage 1 non-responders to placebo and you randomize them to placebo and then whatever your drug is. And there's a statistical way to use essentially all of the placebo and active data. But this way you have some control over it because you already know that you have people who are non-placebo responders going forward into your randomized trial in the second stage. And you can't just ignore what happened in the first phase, as I mentioned. And I'm not I don't have time to go into the details of how you do this, but you can do it in a way that the FDA actually accepts, which is a high bar. And this was studied this was done, this trial, and interestingly, buprenorphine samidorphan did not beat placebo, but the placebo response rate was not that high. I mean, it was relatively modest. It was like 20 to 30%, which is, I think, reasonably acceptable. We don't know for sure that it was caused by this study design, but it has promise, and I think more people are looking at this and other methods. And just one last point before I stop. There's some really simple methodologic things you can do. One is we're wedded to like the p less than 0.05 thing, but the logic of that is actually pretty weak. It's not crazy to say, look, I'm going to do a study and I'm going to power it at p less than 0.10. That's a 10% chance versus a 5% chance. It's still very low. And, it's all about, like, let's say if you're doing something for FDA approval, you go to them and you get agreement or you don't. Or for an early phase study that isn't going to FDA, you can do this, right? It's up to you. You're the one who has to judge whether or not that will meet your needs. And the other is, in many ways, effect sizes are more important than p less than 0.05. Effect size tells you an awful lot. And, it really, you can gauge whether something had a clinically meaningful effect, say. Statistical significance is not clinical meaningfulness. They're different concepts, right? So sometimes, you can do a study and you can decide, I'm going to look for an effect size, and I don't care if I have a p-value, and I'm going to then, if my effect size is greater than something, I'm going to go ahead and proceed with this development, whatever. The other is you can stratify, you can enrich your population to have a better chance of getting better signal detection. I did a study of this years ago where I looked at trials of treatment of insomnia based on their entry criteria for wake after sleep onset. How big of an effect size did they show? Same drug, same dose, or different doses, and looked across a number of drugs. And it was clear the same drug at the same dose had a bigger effect if your baseline wake after sleep onset was higher than a certain amount. And the higher it was, the bigger the effect size. And so you'd be silly not to enrich for a higher effect size. On the other hand, you have to have a population that has clinical relevance. You can't say, I'm only going to look at people who have wake after sleep onset. Uh, sorry, I should have mentioned that WASO is wake after sleep onset, number of minutes awake in the middle of the night. So you have a sleep drug, and you can't. It's pointless to do a study saying I'm going to only look at people who have 3 hours or more of wake after sleep onset. That's a tiny percentage of the population of people who have sleep maintenance problems. So you have to address the practical factors of what's out there, and also it makes recruitment harder and undermines sort of the goal of efficiency. But I'm going to stop there and see if we have questions. Happy to take any questions that people have.